Welcome, everyone. Good morning. Thank you very much, James, for agreeing to give a Build a Cell seminar, and you can get started.
Great. Thank you, Kate, for inviting me. It's always a pleasure to join the community here. So I guess I can give a little introduction of myself. So my name's James. I'm a group leader in Dresden at the Technical University of Dresden. In particular, I'm at an institute within the university called the B-Cube Center for Molecular Bioengineering. And I'm going to tell you today about since I started my group around 8 years ago, trying to understand how membranes work and what lipids do. And in particular, one of the approaches we're taking is working with very simple cells, the minimal cell, for example. And so the work I'm going to talk to you about in the next 35 minutes is motivated partly by two questions, which you see here on the screen. I think you see these, yeah. Why do cells need so many lipids? The cellular lipidomes are very complex, and we have no idea what that's all good for. And another question is, how are membranes genomically encoded, and how can we use that to engineer them eventually? Um, so, uh, my lab, as I said, when I started my group, I was interested in trying to establish new models and approaches to understand the role of membranes, the organizational role of membranes, and in particular, how lipids can organize and modulate bioactivity. And so we're taking two approaches to that problem. One approach is we use minimal cells like Sin3, which are genomically minimal organisms, and we're using them as experimental platforms to study the membrane and eventually hoping to employ them as engineerable membrane systems. And another approach we're taking is sort of a bottom-up approach to ask how lipids can modulate and organize biomolecules and bioactivity. And in this case, we're looking at a sort of unusual interaction, the interaction between RNA and lipids. And I'll tell you a little bit about that work after I introduce the minimal cell work we're doing. So we're interested in the membrane, and we want to study the membrane. We're thinking of the membrane essentially as a hub, a central hub of organization for many crucial processes in the cell, from metabolism to transport and signaling and growth and division. So what you're looking at here is a beautiful animation by Janet Iwasa, showing sort of our canonical understanding of what a cell membrane looks like. You have a lipid bilayer, and within that bilayer, there are a variety of proteins that are either embedded in the bilayer or associated with it peripherally. Now, when the cell builds and maintains its membrane, there are two fundamental challenges it faces. One is ensuring that that 4 nanometer thick oily film with proteins in it is mechanically stable enough to withstand all of the physiological and environmental perturbations that the cell has to withstand, but also that the bioactivity occurring within and associated with the membrane is optimally performing, that's optimized. And the main way that is addressed by the cell is by carefully tuning the physical properties of the membrane, like properties like permeability and bending rigidity and fluidity. And ultimately, the biophysical properties of the membrane are primarily tuned through the kinds of lipids that the cell puts into its membrane. So the structural diversity of lipids and the complexity of the cellular lipidome is essential for tuning and modulating membrane biophysical properties. So in that sense, understanding how life manages complex mixtures of lipids to determine its membrane's physical properties is central, fundamental to understanding the design principles of living membranes. Just to give an example of what I mean, I'm going to show this animation that I had made when I first started my group. It's a very simple cartoon, but I think I like it, so I'm going to walk you through it here. So we can think of the membrane on the one hand as of course, a selectively permeable barrier that lets certain things in and keeps other things out. Now, if we zoom in a little bit, we can see these very crudely depicted proteins. So the membrane can play a role in coordinating the assembly and disassembly of proteins. And there are a variety of mechanisms biophysically that can occur, like phase separation, for example, or curvature is another way that proteins can be localized to certain regions of the membrane. Now, if we zoom into the molecular level, what we see is that it's ultimately lipids that drive these phenotypes. So changing the shape of lipids can modulate curvature, or introducing sterols like cholesterol can promote phase separation. So fundamentally, it's the lipids and their chemistry that the cells use to manage these biophysical phenotypes that are so important to so many processes within the cell. And so the challenge then is understanding why cells have so many lipids. And the reason that's a challenge is because, well, first of all, lipidomes are complex. So you have hundreds of unique lipid species in a typical eukaryote. So here's a log scale depicting sort of the number of lipid species, unique structures in a lipidome. And if we look at eukaryotic organisms like humans, fish, hedgehogs, typical cell in a human or mouse can have 400 different lipid species. There, even microbial organisms like yeast and the bacteria E. coli have relatively complex lipidomes with over 100 unique lipid species. Now, we've identified simpler bacteria, for example, Methylobacterium extercens, which is a plant-associated bacteria we showed had around 30 phospholipids in its lipidome. Um, but still, we're facing a problem in trying to dissect this complexity. Part of it is the complexity itself, but the other part is in experimentally manipulating that complexity. So most cells have very complex metabolic networks that are linked to the diversity of lipids. And reducing lipid complexity involves perturbing those networks, which can get very messy. There's many artifacts that can emerge from genetically perturbing these complex networks. So we're trying to find a way to address this problem where we can explore the role of lipid complexity. And what that requires is a way to essentially tune lipid complexity and minimize it so that we could ask questions like, what were the kinds of chemistries that might have been important for the first protocells and the origin of life? Um, what kind of lipidomes would we need to engineer for membranes for synthetic cells for a variety of purposes, like drug delivery or for bioprocessing? Membranes are very important in many of those processes. So luckily, I was looking for a system that would allow us to really simplify the lipidome and also not only simplify it, but systematically engineer its complexity so that we could study it and understand, the fundamental architecture of lipidomes. Luckily, around the time I started my group, the Craig Venter Institute had produced this organism called the minimal cell, or JCVI SYN3. And this is essentially a bacterium that has a synthetic genome derived from mycoplasma. Mycoplasma are pathogenic bacterial pathogens of mammalian epithelial tissues. So this organism has a synthetic genome with fewer than 500 genes. So this, in comparison to something like E. coli, for example, which has around 5,000 genes, this is already an order of magnitude simpler. One unique characteristic of this minimal cell is that it's amenable to whole genome engineering, meaning that you can really edit you can rewrite the genome to have anything you want. You can introduce new pathways and et cetera. So it's very flexible in that regard. Experimentally, it has a lot of benefits. For example, it has a very simple cell structure. So it's basically a membrane and a cytoplasm, and that's it. So what you're looking at here on the left is this is not actually it's not actually a drawing, it's a simulation of the minimal cell. This is a unpublished work from Zanluthi Schulten and Sievert Jan Marink, where they're modeling the entire minimal cell. And actually, the lipid inset here is based on our lipidomic data from the minimal cell that we reported before. So this cell is so simple that it can be simulated. And that's, I think, an incredible feature because then we can benefit from a lot of the experimental parameters that simulations can access that we can't always access experimentally. Um, it's very experimentally tractable, however. I mean, we can monitor many different biophysical parameters in live cells in the membrane, which is not possible in more complex cells with multiple membranes and other complex features. Finally, one of the most important features of this organism for our question, trying to understand why cells have so many lipids, is that they have a limited, mycoplasma and the minimal cell have a limited ability to make their own lipids. So since the mycoplasma have evolved essentially as molecular parasites of mammals, they've lost the ability to synthesize lipids, and the minimal cell as well. So that opens up an opportunity for us to tune the size and the composition of the lipidome by simply controlling which lipids we feed the minimal cell or mycoplasma in their growth medium. So that was one of the first projects in my lab. Natasha Safronova's PhD thesis um, was essentially to ask if we could chemically define lipid diets that we deliver to the growth medium, and in that way tune the complexity of the lipidome of SYN3. So, for example, we could take fatty acids. If we give them free fatty acids, they can't produce fatty acids and they can't modify fatty acids. But if we give them free fatty acids, they can take them and make a limited number of phospholipids, phosphatidoglycerol and cardiolipin. So by tuning the complexity of the fatty acids that we feed them, we can tune the acyl chain diversity in the phospholipids. We can also feed them, they'll take up sterols or sterile analogs like hopanoids, and we can give them phospholipids and sphingolipids and many other kinds of lipids. So they'll take up just about anything that we fed them so far. And so, in this sense, Natasha wanted to define lipid diets to feed to the cells, and in that way, create cells with lipidomes ranging from low to high complexity, or small to large. In other words, fewer lipids to more lipids. To make a long story short, her project was very successful. So, what you're looking at here is a graph showing how she could tune lipidome complexity, the number of lipid species in the lipidome, from fewer than 10 to more than 100 lipid species simply by tuning the lipid diet complexity. So we have a tunable system that doesn't require genetics and is based entirely on a sort of chemical approach. Our next question from this was how simple can we make, what's the simplest lipidome that can support growth of a minimal cell? And this was the PhD of Isaac Justice. So he built on Natasha's approach, but he simplified it a bit. So he took these lipid diets and he asked, how simple can I make the lipid diet and still have a living cell? So we know that they require cholesterol. We don't know exactly why yet. It's most likely some sort of substrate specific requirement rather than a biophysical requirement, although there is a biophysical element to it. So we gave them cholesterol. And of course, there also has to be a phospholipid that can form a bilayer. So we gave them a phosphatidylcholine lipid called POPC, one type of phosphatidylcholine lipid. So two lipid diet. He got rid of no fatty acids or anything else. So he fed these two lipids to the minimal cell and they grew. So that was good, but we looked at their lipidomic composition. And from these two lipids that we fed them, they managed to generate 28 lipid species. So the minimal cell is trickier than we had anticipated. Well, we were thinking about why is this? So it turns out that the minimal cell has hydrolase enzymes called lipases that can cleave the acyl chains from phospholipids. And then when they have those acyl chains, they can use them to synthesize phosphatidoglycerol and cardiolipin, just as they can when we give them free fatty acids. So we realized that our problem was that the ester linkage that links the acyl chain to the polar lipid head group could be cleaved by these lipase enzymes. So we reasoned that maybe we could trick the cells chemically into only having two lipids by finding an analog of this PC lipid that has a different linkage that they couldn't enzymatically digest. We found a analog on Avanti lipids, which had, instead of this ester linkage, it had a ether linkage. And we reasoned that the minimal cell probably didn't have enzymes for cleaving ether linkages. So Isaac went back to his experiment, but this time he replaced the phosphatidylcholine POPC lipid with a diether analog. He fed those two lipids to the minimal cell. They were growing, but incredibly slowly, like 3 days to each passage, but they were growing. We looked at the lipidome, and 99.9 mole% of the lipids measured from the cells were made up of cholesterol and diether PC. So we managed to simplify the lipidome of a living organism, the minimal cell, down to only two lipids compared with normal cells that have more than 30, more than 100 lipid species. The cells are not happy, but they are growing continuously. We have them growing right now, and it's a fairly robust method. Bachelor students even can go in the lab, and we run a practical sometimes where we have them do this. So it actually works. So they're fine on two lipids. You just have to be patient and let them grow. And so what we've shown with this in a way is we can now say for minimal life, at least, what are the molecular requirements? Of course, we need 20 amino acids, 5 nucleobases, and now 2 lipids. Now, we looked at the cells. We took pictures with electron microscopy to see what happens when we minimize their lipidome. So here you're looking at 3 different varieties of minimal cell with 100 lipid species at the top here, 30 lipid species in the middle, and in the minimal lipidome, 2 lipid species. And these are transmission electron micrographs. And what you're looking at on the left is sort of what they looks like normally. They're kind of ovoid, round cells. But we noticed this other interesting phenotype that emerged with these sort of weird invaginations or holes in the side of the cell, like this sort of, you can see this Death Star phenotype here on the lower right. And when we count the frequency of this phenotype, it increased to about half of the population in the cells with only two lipids. So we were wondering what this was. We teamed up with our colleagues down the street at the Max Planck Institute, Alex van Appen and Petra Kiesel, to do some to generate some cryo electron micrograph tomograms, reconstructed tomograms. So here's a tomogram of one of these cells with these invagination Death Star phenotype. And we see, in fact, that they're actually internalizing membranes. So you have internalized membrane encapsulated vesicles that we think they're being internalized because if we look, for example, at the electron density on the inside of these vesicles, it matches the electron density outside of the cell, which suggests that they're invaginating these membranes, and the inside of those vesicles contains stuff that was in the outside of the cell. So these cells are really unhappy. They're getting really large. I mean, this cell is almost 1.5 microns in diameter, which is 3 times the normal size of these things. They're basically becoming obese with their own membranes. And likely what's happened here is we've removed some of the lipids that are critical for these cells to modulate membrane curvature, for example, cardiolipin. And so probably this phenotype is a result of the fact that we've disrupted their ability to modulate membrane curvature, and this is causing all these aberrations in membrane invagination. So this is something we can now begin to explore how to restore that phenotype and which are the specific lipids that are important for this kind of regulation. But for me, one of the most exciting outcomes of this observation is that half of the cells are still perfectly fine. They're round and they're growing normally. So even with two lipids, the minimal cell might be slow, but they're chugging along. Okay, so what have we achieved with this? What we have now is an experimental platform, a living cell in which we can tune the membrane composition to have lipids from two lipids to more than 100 lipid species. Not only can we tune the lipidome size, but we can also tune the chemistry of the lipidome to reflect membranes that are more bacterial or more like for example, the plasma membrane of a eukaryote with cholesterol and sphingolipids. So we have a very flexible system in which we can now begin to systematically ask in a quantitative way why so many lipids are important for membrane biophysical phenotypes and for cellular fitness. Just an example of what this could be useful for. I mentioned we can tune the membranes to have features that are more like bacterial or eukaryotic. And one way to look at that is the surface charge of the membrane. So we can feed the minimal cell lipids that make the membrane have a negative surface charge, for example, when they only make PG and cardiolipin. And this would reflect more typical bacterial membrane. Or we can give them lipids that are zwitterionic, that are neutrally charged, and get a membrane with a surface charge that's neutral. And this is interesting because then we can think about asking questions like, for example, screening for antimicrobial peptides. So the idea here is that there are small cationic peptides that are active against bacteria because they have a positive charge and they interact electrostatically with the bacterial's membrane's negative surface charge, and they bind and then form pores and do a lot of other things downstream of that. But the reason they're useful as antimicrobials is because they don't interact with eukaryotic membranes, because the eukaryotic membrane is neutral and there's not a strong electrostatic interaction to engage the peptide with the membrane. So we've just done a proof of principle showing how we can use the same cell with two different lipidomes to screen for this interaction using a very well characterized peptide called gramicidin. So we can see on the left here, these are cells grown with just cholesterol and fatty acids, and they only have negatively charged lipids. And the introduction of even incredibly low nanomolar concentrations of gramicidin kills them. On the other hand, if we grow them with these neutral membrane surface charge, they become much more resistant, up to hundreds of nanomoles of gramicidin. So this is potentially a nice screening platform for antimicrobial peptides. We could even take this a step further and use it to screen for drugs against if we heterologously express enzymes like single lipid modifying enzymes in mammalian cells that are associated with disease. We could identify drugs that might inhibit them or activate them. So I think this is an exciting application for this approach of tuning the lipidome. Okay, I want to switch gears a little now and tell you about something, a different part of the lab that I mentioned at the beginning of the talk. And this involves the work I told you about, where we're trying to understand how lipids can interact with and regulate the activity of RNA. This was originally motivated as a kind of an origin of life project. We were interested in the RNA world, the idea that RNA, because it can both catalyze chemical reactions and contain heritable information, that maybe it was the important molecule in the first life forms, because it's a simple solution to solving the problem of heredity and catalysis. And of course, one problem is how do you organize an RNA world? And we thought that maybe introducing lipid membranes to the RNA world could be one solution. And the idea was that if there were direct interactions between RNA and membranes, that the membranes could provide certain advantages or benefits to catalytic RNAs. For example, constraining them to the membrane surface would increase local concentrations and reduce dimensionality. All of those things will enhance the activity of the interactions of RNA ribozymes, catalytic RNAs, and their substrates. So we were really excited about this. And so, of course, a question emerges, do RNA and lipids interact? And this was a problem that Michael Yaris and his colleagues addressed over 25 years ago in Colorado when they engineered synthetic RNA aptamers. So aptamers are short RNA sequences that are generated through sort of in vitro selection. And they have a high affinity for a particular target. And usually that target is a protein that you select for. But in this case, Michael Yaris and his group asked if they could make aptamers for phospholipid membranes, and it worked. And what you're looking at here is an image that we did just to confirm, to reproduce those results, where you're looking at a giant unilamellar lipid vesicle. The scale bar is 5 micrometers. And it's made of 3 different kinds of lipids that allow it to phase separate into 2 liquid phases, an ordered and disordered phase. And the disordered phase is shown in silver here. It's labeled with a fluorescent probe. And the ordered phase is actually shown in green, is actually labeled with a fluorescently labeled RNA aptamer, one of the aptamers that Michael Yaris published 25 years ago. So what you see here is the astonishing observation that RNA can actually bind selectively to phospholipid membranes. Not only can it bind to phospholipid membranes, but it can select membranes, they can be selective for membranes with a certain property or composition. So this has a lot of exciting implications, not only for the RNA world and origin of life scenarios, but for synthetic biology, for thinking about how we can use this very simple biomolecular system, lipids and RNA, to organize, to localize RNA or to regulate it. And so just to give you an idea of the selectivity of this interaction, here is another phase-separated membrane. We have an ordered and disordered phases. The ordered phase is in this dark patch where the probe is excluded. And we've added some random, I think these are probably 40 nucleotide oligos randomized, stained with cybergold. And we can look at the merged image, and you see how the RNA really localizes to these ordered domains. This is a reversible process. So, for example, here we can heat the system up slowly, and at some point we'll cross the phase transition temperature boundary. And at that point, this ordered phase will demix into the bulk phase, and the RNA will then delocalize from the membrane surface. And it's reversible. We can go back down and it'll relocalize. So with something as simple as just tuning phase properties of the membrane with temperature, we can control the localization of RNA. So that's pretty cool. Now, I don't want to I don't have time today to go walk you through another entire story, but I want to just give you some highlights from this first paper we published several years ago. It was the PhD project of Thomas Cherniak. And the title pretty much says the punchline that we showed that lipid membranes can modulate the activity of RNA through sequence-dependent interactions. If you take a picture of the QR code, you can get the paper. And I just want to point out one interesting insight from that paper that I think will be interesting in the long run for our goals of studying membrane and architecture in the minimal cell eventually. So we were looking at how sticking a RNA ribozyme, they call the R3C ligase. It's a ligase ribozyme. How sticking that to a membrane could alter its activity. And in the end, we modified the system. We modified the substrate, RNA substrate, in a way that it bound the whole system bound to the membrane together, the substrate and the ribozyme. And in doing that, we sort of by accident created a situation where the ribozyme activity was rescued essentially, or activated by the introduction of membranes. So this modification we made to the substrate, we added some additional bases, some guanines that allowed it to stick better to the membrane, made it stick better to the membrane, but it also inhibited the ligation reaction by causing the substrate to fold over and become inaccessible to the ribozyme. But when we introduced membranes, you can see in this figure here, you're looking at the reaction rate on the y-axis of the ligase reaction, and on the x-axis is increasing concentration of lipid vesicles. What we showed was by introducing lipid vesicles, we could rescue the activity of this ligation reaction. And the reason that happened is because the vesicles would interact preferentially with this modified part of the substrate, allowing it to unfold and then allowing the ribozyme to activate it. So this to us was really exciting because it sort of showed a proof of principle, at least, that a riboswitch that's sensitive to lipids and membrane property could potentially be engineered and may potentially exist in cells. They just haven't been discovered yet. And so that opened up an interesting thought that maybe we could use lipid-sensitive RNAs as a component to engineer lipid homeostasis pathways by constructing riboswitches. Riboswitches are RNA sequences that are usually found in the untranscribed region upstream of a gene. And when they interact with a molecule or respond to something like temperature change, their structure changes. And that allows them to inhibit or promote translation of that mRNA. So that's one idea that we're thinking will allow it will ultimately merge these directions we're exploring using the minimal cell to study membrane design principles and RNA lipid interactions potentially to engineer sort of orthogonal homeostasis pathways. Okay, we just published a new paper this year where we extended this observation to naturally. So the R3C ligase is an artificial ribozyme that was generated by Gerald Joyce and his colleagues at the Salk Institute. We've now extended these observations to naturally occurring ribozymes like hammerhead and HDV, and showed similar effects of lipids on their activity. Okay, so what have I shown you here? Um, we've established an approach to tune the lipidomes of mycoplasma in the minimal cell, SYN3. We've used that approach to minimize the lipidome to only two lipids, demonstrating that two lipids are sufficient, but barely sufficient for life. And we've shown that lipids can actually interact with RNA and modulate their activity, and this opens the path for designing lipid-sensitive riboswitches. Okay, so I want to give you in the last few minutes here a little insight into, how we're thinking about this problem of lipid complexity and engineering membranes. Let's come back to this question of why are there so many lipids? Life has evolved diverse strategies to adapt to different environments, and lipids are part of that strategy. So, for example, ctenophores in the deep ocean. There was a recent paper by Ite Budin showing that these deep sea organisms modulate lipid shape, the geometrical shape of lipids to respond to the challenges of the high pressure in the deep ocean. Reindeer, interesting example. They walk around in the permafrost, and of course they're mammals, so they're 37 degrees or around there, but their feet are really cold. So actually, their cell membranes in the extremities have really high concentrations of polyunsaturated lipids to compensate for the rigidifying effect of the cold temperature in the membranes. Archaea are well known for their ability to adapt to extreme conditions, and one of their lipid innovations was these membrane-spanning lipids that create a monolayer essentially and make the cells very resistant to a lot of chemical and physical stresses. We showed with work when we collaborated with Diane Newman that hopanoids, these are sterile analogs, are really important for certain nitrogen-fixing bacteria to withstand the sort of osmotic stresses involved in colonizing the root nodules of plants. So, why has life evolved this huge diversity of lipids, and how can we use that to engineer membranes in life? How can we use that knowledge? So, one current unknown or problem we're trying to understand is precisely how does the complexity and composition of lipidomes determine the physical properties of membranes. And we're thinking of these physical properties in a way as phenotypes of membranes, like permeability and curvature. These phenotypes are derived from the molecular composition of the lipidome. And in turn, they have pronounced important consequences for organismal fitness. So the physical properties of the membrane, like, for example, permeability is very important for metabolism. If the membrane is leakier, the cell has to invest more energy in maintaining ion gradients that are important for energy metabolism. Curvature, fluidity, these are all physical properties that have consequences for organismal fitness. And so, of course, we would assume that they're acted on by natural selection through evolution. So what we're trying to do is to take advantage of this minimal cell and our ability to tune the lipidome from a minimal lipidome of two lipids to more complex lipidomes to begin to systematically explore what's the molecular basis in terms of the lipidome of membrane biophysical phenotypes. So, in the minimal cell, we can not only tune the lipidome, but we can also measure a variety of biophysical properties in vivo in living cells, like viscosity, lipid order, permeability, phase properties, things like that. And in turn, we can look at how those are influenced and are correlated with different fitness parameters like growth rate and cellular robustness, antibiotic resistance, the viable growth range, like temperature over which it can grow. So we can begin to try to piece together a picture of how the molecular complexity of the lipidome influences membrane phenotypes and organismal fitness. Um, of course, it's also important to think about how the lipidome is regulated, right? Metabolism, the cellular metabolism acts on the lipidome to ensure that they have the right lipids to create the right biophysical phenotype so the cell can do its job properly. We published a paper last year where we demonstrated that both Mycoplasma mycoides and JCVI Syn3b are capable of adapting their membrane properties and consequently their lipidome composition to temperature change. And so this raises a question: how does such a simple minimal organism achieve such complex regulation of its lipidome? And this is one problem we're beginning to try to understand in the sense of, what are the pathways and mechanisms that allow these minimal organisms to adapt, to sense and respond to environmental perturbations and to regulate homeostatically their membrane's properties. So we're thinking about it kind of like as a system, like involving sensors, controllers, and effectors and response. And there is emerging work showing that organ cells have dedicated sensors in their membranes. They're proteins that sense physical properties of the membrane, and they then in turn regulate lipidome composition. So this is one possibility in the minimal cell, that there may be dedicated membrane sensors that we could discover. Alternatively, it might also be the case that there's sort of more collective sensing of the membrane proteome that somehow feeds back into lipid metabolism. And these are directions we're beginning to explore. Um, Not only does the cellular metabolism regulate lipidome composition, but the lipidome composition and the membrane's properties dictate or influence strongly the cell's metabolism. And so one way we're beginning to explore that is I'm collaborating with Karim Fahmy. He's a physicist, a biophysicist in Dresden. And we're using microcalorimetry to very precisely measure the heat generated by the minimal cell so we can generate these heat flow curves. And Kareem together, we're working on a paper together where we can show that metabolic heat flow from the minimal cell is very sensitive to the lipidome composition. And changing the lipidome composition determines how energy is distributed between biomass forming and non-biomass forming processes. And we can sort of think about trying to understand that in the context of fitness landscapes. So we're very interested in understanding not only how does metabolism regulate lipidome composition, but how does the membrane's composition influence cellular metabolism. So, okay, coming back to our problem, we ultimately, if we want to engineer membranes, we need to understand how their design principles. And of course, we can do things like I mentioned at the beginning of the talk or towards the middle of the talk, that we can tune the lipidome composition in that way, make cells with more bacterial eukaryotic properties through a purely chemical approach. But if we want to extend the function of the minimal cell membrane, to engineer the membrane for new purposes and functions, we ultimately have to be able to understand how it's genomically encoded. So for membrane proteins, that's not a problem. We have the central dogma: gene, mRNA, membrane protein. But for membrane biophysical phenotypes, it's a much more complex relationship between phenotype and genotype. So we're interested now in the long term in understanding what are the genomic determinants of membrane phenotypes. And this really requires taking a holistic view of all of these interactions between metabolism, the lipidome, membrane biophysical phenotypes, and of course, organismal fitness, and how that's ultimately determined in the genotype and how we can eventually program it or engineer it through genome engineering. And for example, not only would this allow us to engineer membranes, but it would also allow us, if we understand the genomic determinants of membrane phenotypes, we can actually begin to reconstruct their evolution. And I've teamed up with some computational biologists Anflo Bitball and EPFL and Edward Lyman in Delaware. And one of the ideas is we want to try to start reconstructing ancestral lipid genes and pathways and then reconstituting them in the minimal cell and using the minimal cell essentially to resurrect ancestral membrane phenotypes, and in that way, sort of perform a kind of molecular paleobiophysics, study the physical properties of extinct membranes. So that's one thing that understanding the genomic basis of membrane phenotypes would allow us to do. So, okay, just taking a step back and giving you a final take-home message from all of this. What we're ultimately in the long run interested in doing with all this work I've told you about today with the minimal cell is to try to gain an understanding of how membrane phenotypes and their regulation are genomically encoded so that ultimately we can engineer them for synthetic biology. Great, thank you very much. That's all I have for today. I have to, of course, thank my lab. They do most of the work, and the Technical University of Dresden and BCube for hosting me. Our funding, Lipotype, has done all of our lipidomic analyses, and many friends and colleagues that have helped me along the way. So with that, I'm happy to take questions. Thank you.
Yeah, that was fantastic. I always like your talks because it kind of challenges the
Thank
way
you,
I
Kate.
think about lipids, even though I'm a lipid person to begin with. There's plenty of questions in chat. Can you see chat?
Let me pull it up here. Yeah, I got it now.
Can you read a
Okay.
question out loud because people watching the recording don't see the chat?
Sure, I'll just start from the first one. So, Abby Grebson writes, when you tune the lipidome using media lipid composition, how long does it persist? For example, if you change media, how many generations do you see the change? Good question. So, for this, I mean, it persists as long as we continue feeding them, growing them in that media. If we change the lipid diet, for example, which would be relevant to your question, it usually takes about 2 to 3 passages. In other words, a passage is when we inoculate, transfer cells from a dense batch culture to a new batch culture. It takes about 2 or 3 of those passages before the paternal lipids from the former lipidome are diluted out. So in that sense, yeah, that's about the time scale of changing the lipidome. If you have another follow-up question, just write it in the chat. Second question, Luis Daniel Cruz Zaragoza. How many division cycles can the syn-3 diether PC2 lipids go through? How long does it take for the disappearance of lipid complexity in that system? Okay, that's a similar question to the previous one. When we introduce the 2 lipid diet, it takes about 3 generations before we see the more complex lipids diluted out and we only have 2 lipids. Third question, Sienna Melbourne. Did you identify the enzymes that Syn3 used to process the ester bond and the lipids you originally fed it? Not yet, but we're very interested in trying to identify those because A, we can get rid of them and that would simplify our life a lot, but also it would be very interesting in general to understand the pathways that the minimal cell has left that it can use to modulate the lipidome. Okay, next question. Ranjan. What are the molecular interactions of RNA and disordered lipids, which is missing in ordered lipids for lipid RNA interactions? Is it solely ionic interactions? Okay, very good question. I didn't actually go into much depth there, but it is primarily an electrostatic interaction that's dependent on divalent cations like calcium and magnesium. And there's a number of computational studies that have followed from our work in 2022 that indicate that there's a cationic bridging, divalent bridging between the phosphates of the RNA and the phospholipid groups. But they're still leaving open the possibility that there may well also be hydrophobic interactions and, of course, entropic factors of the interaction of, for example, an RNA that's rigid with a long persistence length and a rigid membrane can also play a role. Okay, next question. Henrika Wertheimer. Did you ever try to go the other way and see how complex you can make the Syn3 lipidome? How many types of lipids geometry can you stick in there? Great question. Not well. So we've reintroduced complexity to the 2 lipid lipidome, and that's in the Isaac's paper. So we systematically tried to, for example, introduce more acyl chain diversity or more head group diversity independently. And there's some limitations to the way we did that approach. I would love to refine it a bit more, but the first order result we got from that set of experiments was that, in fact, the diversity of acyl chains could rescue growth, whereas the diversity of reintroducing head group diversity couldn't. So that's an interesting clue that acyl chain diversity seems to be, at least in those very minimal lipid mixtures, more impactful than the diversity of the head group chemistry of the lipid. But for sure, it would be interesting to go further and start to really explore different complex lipidomes. And at what point is the benefit no longer, do you saturate the benefit of complexity? And just my guess is probably around 20 to 30 lipids, but I don't, that's based on a few very loose experiments. But yeah, that's a very cool question. Okay, another question here. What other factors, in addition to the genes, would you consider for the shaping and function of ancient membranes? Ah, yeah, good question. I mean, of course, the genes, the genome interacts with its environment, or the product of the genome ultimately interacts, is dependent on the environment as well. So I think we have to take into account the cellular composition, that's an environmental factor, as well as the kind of environment that the cell's growing in, because all of these things will ultimately shape um, the what that genome can the emergent properties of that genome's products. So I think we not only have to think about the genomic determinants, but more broadly what the cell's made of, what pathways are there, how they interact, the emergent properties of that system, as well as how that interacts with the external environment. One last question here. So, can you do the same lipid diet to more complex cells like E. coli? That's a very good question. So, one of the reasons we went to the minimal cell is because it is a lot more challenging in an organism like E. coli to do this because, first of all, E. coli isn't competent to take up lipids like minimal cell can. And so, the main way that you would tune the E. coli lipidome is through genetics, like, for example, knocking out certain pathways or introducing genetic ways of tuning different pathways. And there's some very nice work that's done that. Like recently, Ite Budin showed with yeast that they could tune membrane fluidity by genetically tuning the number of double bonds on the phospholipids. And they did some very nice work showing how fluidity is super important for respiration because it limits the diffusion of electron transporters. But you can't get anywhere close to the level of control that we have in the minimal cellular mycoplasma through that sort of genetic approach with something like E. coli. Another problem with E. coli is it's a gram-negative bacteria, so it has two membranes, an inner and an outer membrane. And that makes it more experimentally difficult to measure different biophysical properties of the membrane in vivo, because usually the way we measure those properties is through some kind of fluorescent probe that responds to the environment in some way or another that we can measure. And fluorescent probes typically just go everywhere where there's a membrane. So you end up staining inner and outer membrane and you don't get a clean signal. Whereas with the minimal cell, there's only one membrane. So that's the only thing that's causing us any trouble. So E. coli is better for a lot of other reasons. It's easier to grow. It's more, there's in some ways more genetically tractable, but the minimal cell is the only organism that we can do this kind of work with right now. So
Right.
that's the end of the chat
Yeah,
questions. If there are any more
I
from
think,
the
yeah, I think you went through all of them. Yeah, I would be really curious to see stuff like that, not even with E. coli, but like with an non-minimized mycoplasma.
yeah, yeah. We do that with, I mean, we most of the work we've done so far, we've done in parallel with Mycoplasma mycoides. Um,
Yeah.
and actually, a new organism we're exploring is Mesoplasma florum. This
Oh,
uh, this is also a mycoplasma-like organism that's probably an insect-associated bacteria. It's almost as minimal as SYN3. It's naturally occurring. It doubles every 30 minutes, so it's really fast. And it has some features that are great for us. Like, for example, we can, with the minimal cell or mycoplasma, they always need cholesterol. So if we want to explore the role of like bacterial cholesterol analogs like hopanoids, they will grow on them, but there always has to be cholesterol there as well. Mesoplasma can grow only on hopanoids or only on cholesterol. So then we have a system where we can really directly compare the chemistries of those very important ordering lipids.
and I think Luis has one more question.
Ah, yeah. What happens to the membrane transporter structure and function in SYN3 diether 2 lipid cells? I would love to know. I mean, this is a very good question. And I mean, this is something that another feature of the cell that I think will be really interesting is the possibility that we might be able to, with some careful cryo-EM, get the structures of membrane proteins in vivo and then tune lipid composition and ask how the lipid environment influences the structure and conformation of membrane proteins. I mean, of course, that's not trivial. I mean, working with getting structures with a live cell in the background and all the noise will be tricky, but I think that would be a very exciting thing to pursue.
Thank you.
Yes.
Thank you so much.
Absolutely.
Okay.
My pleasure.
If there are no more questions, I imagine people know how to track you down if they have more.
Yeah, you
And,
can always just Google me, send me an email. I'm always happy to chat about these things.
and thank you so much, James, and thanks everyone for great questions and great discussion.
Thanks a lot, Kate.
And have a great rest of your Monday.
Yeah, you too. Have a
Bye.
great week. Take care.
Bye.
Bye-bye.